Hola. Hola. Está. Olá, boa tarde. O professor Eduardo está sim. Sim. O, isso, o professor Yunex. Olá, boa tarde. Sim, claro. Listo? Pronto? Pois. Uhum. Pois. Okay. Well, first of all, thank you all for being here. As all the members of the jury. I will start with my presentation. Okay. Okay. I can begin, right? I can start. Okay. So. Okay. Okay. So. Welcome to my presentation. This is my thesis. The title is Machine Learning Models for Predicting Disease-Related Biological Interactions. I am Iván Carrera. I'm a professor of the Department of Informatics and Computer Science of Escuela Politécnica Nacional here in Ecuador. My advisors are Inés Dutra from the Department of Computer Science of University of Porto and Eduardo Tejera from the Group of Chemobioinformatics of Universidad de las Américas here in Ecuador. My presentation has three parts. First, an introduction about the problem, motivation, the state of art, and my proposal. Second, the presentation about the methods that were developed in this thesis. And lastly, conclusions and future work. Okay, so let's begin with the introduction. Before I speak about this work, I should talk about the concept of drug repositioning. Here we have this model that represents this graph that represents the interactions or the associations between drugs and targets. Targets being all the biological entities to which a drug can be aimed at. So, drug repositioning is the process in which we discover new associations between drugs and targets. Okay, in this work we work with targets as cellular lines. Okay, so here in this example we have drug X, and this drug X is similar to drug 2. Since we know that there is an association between drug 2 and target B, we can predict based on the similarity principle. Given that drug X is similar to drug 2, we can predict that there will be an association between drug X and target B. Okay, so this is what is called drug repositioning. Similarly, we can say that another type or another kind of drug repositioning is working with the similarity between targets. Okay, so we have this drug that it has an association towards target B. Okay, so given that there is this target X that it is similar to target B, we can predict that there will be an association between drug 2 and target X. So, but this presents two challenges. First, the definition of the representation of what a target is, and also what it means to be similar. What is the measurement to the similarity between targets? Okay, so these are two problems that we will address in this thesis, in this presentation. Okay, so the motivation is that cellular lines are an important tool for cancer research, and computational models of cellular lines can optimize in vitro assays in the drug discovery processes, especially in the drug repositioning process. A good characterization and representation of cellular line is important to allow for automatic discovery of cellular line interactions. The drug repositioning models can have two approaches. First, based on sensitivity profiles, and second, based on gene expression of cellular lines. Okay, so the gene expression is by a majority the most used model for the representation of cellular lines. But gene expression presents a problem that these methods require the gene expression profile. And this gene expression profile is triggered by the compounds, meaning that for a different compound applied to a cellular line, you can have two different gene expression profiles, okay, depending on the compound that is applied to the cellular line. Okay, so this makes difficult for a general representation, because this representation for gene expression depends on what is the compound that is applied to a certain cellular line. Okay, so there's a need for a drug repositioning model for cellular lines, which can rely upon a general enough representation method that can be utilized with a large number of cellular lines. The computational models for drug repositioning for cell lines, okay, they are mostly based on collaborative filtering and recommender systems in matrix decomposition, which are all very similar. Okay, so we could also base our work into these types of work, and we can perform the discovery, the repositioning for the cellular lines based on this type of works. Okay, however, all these works are based on specific databases or specific diseases. So this is dependent on the models. There is no one general model for all the cellular lines, but all these work for only a specific data set or a specific disease. So our proposal is to have a very comprehensive approach for the prediction of the drug cell line activities. The strategies for the drug repositioning for cellular lines are, according to Dudley et al., drug-based or disease-based. Okay, so the type of works are discriminated according to these approaches. Okay, so they give and keep and pour divide into two categories. First, network-based, and second, learning-based. Okay, and in learning-based, they divide into two categories as well: feature vector and literature analysis. Literature analysis meaning that all the information that is recovered for these methods is based on an analysis for the literature on the what are the, for example, what are the interactions that are written in the literature, and we will base our work into these two categories. First, a literature analysis, because we will explain about the proposal in which we propose to make an analysis of the literature and also to represent cellular lines based on feature vectors. Okay, so even though all these methods are used in the scientific literature, none of them is used for a more comprehensive approach for describing a large set of cellular lines. Our proposal is to have a general approach for describing cellular lines. Okay, so what do we propose? First, a method for automatically characterize cellular lines from the scientific literature based on support vector domain description. Okay, and second, a set of 4 methods for drug repositioning for cellular lines. The first is based on molecular similarity, the second in cellular similarity, the third on affinity profiling, and the fourth is a consensus model that integrates the former 3 methods. Okay. The contribution is to be understood into these 2 fields. First, biocheminformatics, and second, computer science. First, in the field of biocheminformatics, we will have a representation model for cellular lines, and second, a novel methodology for drug repositioning for cellular lines. In fact, 4 novel methodologies for drug repositioning for cellular lines. Okay, and in the field of computer science, first, a representation model for complex entities. The application here is cellular lines. And second, a novel methodology based on consensus for link discovery. In this case and in this way, we can discover the associations between 2 types of entities in bipartite graphs. Okay. So I will present now the 5 methods. Okay. First, the methodology for cellular line representation. Okay, here we have the pipeline that we use for the characterization, for the representation of cellular lines. Okay, so first of all, we will identify which are the cellular lines that we will represent using this methodology. Okay, so once we establish the set of cellular lines that we will represent, we retrieve the abstracts from the literature, all the abstracts that are related to these cellular lines which were identified. Okay, then we form our corpus. Okay, all the corpus, all the set of abstracts that relate to these cellular lines. Then we transform this data, this corpus, into TF IDF vectors. Okay. Afterwards, we perform a feature selection algorithm, and in these two results are to be understood like this. First, we perform a document classification algorithm. This in order to understand if it is possible to differentiate between classes, between cellular lines. Okay, using the information from the abstract that are related into these classes, into these sets of cellular lines. And if we were successful, which we were, perform a cell line description okay, using this representation. Okay, so for the document classification algorithm, okay, we saw that there was this dependency for the performance for the minimum number of documents. This means that once we have all the abstracts that talk about the cellular lines, okay, we saw that there were some cellular lines that had more documents and other that had less documents. Okay, so if we only select the cellular lines that have, for example, at least 10 documents, okay, so the performance of the document classification algorithm is in this case poorer than if we select only the cellular lines that have at least, for example, 20, 30, 40, etc., up until 100 documents. Okay, so this means that if you have more information to represent cellular lines, the representation will be better. Okay, however, this method is not as general as you wish, as you would like to. Okay, because if you only select the cellular lines that have at least a certain number of documents, okay, you will select a smaller set of cellular lines, okay, if you have more minimum number of documents. So we established that with 60 documents at least, we have for the test dataset a precision of 70%, which was what quite enough or sufficient for us to perform this cell line representation. Okay, so the cellular lines are represented like this. Okay, so this cellular line, for example, this is an example. These are the word counts for the TF-IDF algorithm for the first word of the vocabulary, for the second, for the third, so on and so on and so on. Okay, and with this representation, with these points in the space, we calculate a sphere. Okay, so this is the sphere representation of domains as was developed by Tax and Duin in supervector domain description. So in this case, all the documents that are related to cellular line 1 here, okay, are enclosed in this sphere. Okay, this sphere is characterized by its center and its radius. Okay, for in this case for cellular line 1 and in this case for cellular line 2. Okay, so we measure the distance and we measure the intersection between these two cellular lines. Okay, this is the formula for the intersection. In this case, the intersection is negative because these are disjoint cellular lines, meaning that there is no document that is common for these two cellular lines. Okay, in the other case, for having intersecting cellular lines, the intersection here is positive. Okay, and this area here is all the works that are related to cellular line 1 and cellular line 2. Okay, so this is the intersection. Okay, for this special case for tangent cellular lines, which wasn't very common, okay, the intersection is 0. Okay, meaning that these cellular lines are adjacent, but there is no commonality between them. Okay, and here the circumscribed cellular line, in this case the intersection here is the diameter of the cellular line 2 here, meaning that all the works that are related to cellular line 2 are included in the set of works that are related to the cellular line 1. Okay, so this is the representation of cellular lines. Okay, now the methodologies for the repositioning. The first work that we published was this predicting activities between cellular lines and compounds based on molecular similarity. Based on molecular similarity, meaning that what it was called the self-fishing algorithm. Okay, as I said, the drug is similar. The drug in that we query is similar to drug 2, and if this structure has a known association, we can predict also a known association. The self-fishing algorithm, okay, defines two strategies. The first strategy was to the most similar drug, and the second strategy was with the top three, with the top three most similar molecules, okay, most similar drugs, and with this set of three molecules, we inherit the associations with these targets, okay. Here are the results of our approaches as we were trying to obtain the best parameters for this approach. We tested two approaches, as I said, S3 and S1, with the top 3 and the top 1 similar molecules. We also explored the efficiency for the similarity cutoffs for the representation of the molecules. Okay, the best one was with 0.55. In here we define that the best approach is S3. Okay, and also with the representation with radius 8 for the molecules. Okay. Second, the second model is called compound fishing. Okay. Also, we detect the activities between cellular lines and compound, but we are based on cellular similarity. So, based on the representation of cellular lines, we establish what is this representation, what these targets mean, and what is the similarity, the intersection between the spheres of the representation for the cellular lines. Here, we also took the top 3 most similar cellular lines, and we inherit the interactions for these 3 most similar cellular lines. Okay, we only tested the S3 approach since we already tested in the self-fishing algorithm. Okay, and also one change that was introduced here was that we tested with all sensitive and resistant activities between cellular lines and compounds. Okay. The third model that we developed was collaborative filtering. In this case, we formed an interaction matrix between drugs and cellular lines. This interaction matrix doesn't have information about cellular lines nor the drugs, only the information which was called the affinity profile. An affinity profile for a drug is a set of cellular lines that it can relate, and the affinity profile for a cellular line is a set of drugs that it can relate. So, using a collaborative filtering algorithm, okay, we can have these new values, these recommendations for the new interactions between cellular lines and drugs. Here, this interaction matrix in blue, what we did was this. If the drug was understood to be active to the cellular line, or the cellular line was sensitive to the activity for the drug, we establish a number of +1 here in this cell. And if the cellular line was resistant to the activity of the drug, or the drug wasn't active to this cellular line, we put the number -1 here. If we do not know what the activity was between the drug and the cellular line, we establish a number 0 here. The collaborative filtering algorithm, what it does is to calculate the number 0 to not be 0, instead a number between -1 and +1. So if we can understand this estimation, this number, we can also predict that there will be an association between a drug and a cellular line. Okay, so we tested, for example, with database Campbell versions 28 and 29. We train our model with version 28, and we take the new records from version 29, and then we assess if our model is capable of predict these associations. Okay, so the area under the ROC curve was 0.8. which is a pretty good performance. Okay. We also had to understand or calculate a threshold. Okay, so the values for the predictions, for the estimations, are between -1 and +1 here. Okay, so what is the best threshold in terms of performance to be to the estimations? We established that the threshold is -0.43 here. So if the value is lesser than -0.43, this estimation is understood to be -1, to be a resistant cellular line to the drug. And if it is bigger than -0.43, we understand that it is a +1, so the activity is to be understood as sensitive. The cellular line is sensitive to the drug. Okay, and now we combine these three models. Okay, these three models, self-fishing, compound fishing, and the recommender system, were combined into an ensemble method based on a supervector classifier. Okay, having a better performance in terms of precision and accuracy. Okay, we took this supervector classifier and compare with a voting method. This voting method was very simple. We since we have three models here, so we perform a simple voting method. If two of them classify an interaction as a +1, as an activity, and the other one says as a -1, as an inactivity, so the majority says that it is an activity. So this is basically the voting method. However, the precision and the recall and also the accuracy was poorer than the ensemble method, because the ensemble method gives in every case gives a relevance or an importance for a specific model. Okay, so we can benefit from the combination of this model, from the consensus of these three models. Okay, so the conclusions are here. The objectives of the work was to develop a set of models for discovering associations between drugs and cellular lines, and also to represent cellular lines. We have accomplished this objective in a few steps. Our models make a contribution in the representation of entities and the representation and discovery of relationships. Our model for cellular line representation can be applied to a large set of cellular lines, provided that each cellular line have a sufficient corpus. Our model for the discovery of interactions between cellular lines and drugs have an important performance. Our models feel the need for a general enough drug repositioning method for cellular lines. The works that were published were these five. These three are papers. The first one was published in BIBM, the second in the +1 journal, and the second in the Health Informatics Conference. Okay, this one for the representation method, this for self-fishing, and this for the collaborative filtering algorithm. Also, I published a poster for the representation it was a proof of concept of the representation of cellular line, and a summary of this thesis was presented in the doctoral consortium of Biotech in 2021. Also, a related work that was I advised with the supervision of Dr. Ines Dutra was a master thesis with title Drug Repurposing with Association Rules, presented by Sara Rocha. okay, from the Master of Bioinformatics and Computational Biology of University of Porto. And currently, I am advising a graduation project in Politecnica Nacional with title Computational Performance Analysis of Recommender System in Pharmacological Databases. Okay, the further work, the work that has to be done is first to improve and expand our models. Okay, there's always room for improvement and expansion of models. Second, the exploration of the representation model for different entities, not only cellular lines. Third, to address the cold start problem for our recommender system. Okay, the cold start problem is when we have a new entity and we do not have much information about that. And we have to address what to recommend to a new user in a system. And fourth, they explore the possibility of computing an ontology, okay, of cellular lines based on our representation and compare these to other ontologies that are published in the literature. Thank you very much. Thank you.
Okay. Well, we already discussed that with you, Iván. I missed in the document some examples of any computer guided repositioning effort that have worked in the clinic. I know there are a few of them, so I suggest that once you finish or make the modifications in the document, you can include some of them. Also, to be clear in the document in the difference between a ligand and a compound.
Yes.
A ligand, yeah, we talk about that already. A ligand is when the compound already binds a target. So not every compound is a ligand, and then you have to clarify that in the document. Also, in the concept of QSAR, QSAR is not only used to model structurally related compounds. You can also have QSAR models for data sets which are diverse in terms of structure. What else? For target fishing, you need usually not only the ligand information, but also the ligand target interaction information. So you can train or prepare your models. That's something that was missing in the document also. Well, in vitro screening also to correct that the molecules are evaluated with the aim of identifying bioactive compounds. I mean, you don't test bioactive compounds in virtual screening, but you attempt to identify these compounds during virtual screening. I could also you to clarify that virtual screening is not always phenotypic. You can have virtual screening focusing on specific targets too, and that is beyond the phenotypic virtual screening. Also, in some works that you discussed on previous work that you discussed, where some experiments were performed, it could be interesting to have in the text which hit rates were obtained for these experimental validations. So this is something that would enrich the document. Also, the TF-IDF abbreviature, please define that
Okay.
in the document because it's missing. So I couldn't understand. I mean, I understood that that was a vector, but which kind of vector and so on, it was not clear for me when I read the document for the first time. Also, to be more clear on section 4.4, that feature selection was based on a variance filter. So the link was a bit missing, at least for me there. And also in the title of chapter 5, I consider, it's up to you, that interactions is more appropriate than activities. Instead of predicting activities, predicting interaction, because your endpoint is not a continuous endpoint, it's true, false, or no information, okay? And also that you mentioned in section 6.4 that there are 4 versions of Kimball database. However, you list 5 versions. I think that this is an update program that you forget to change the number. And please also to explain the terms in equation 6.1, 6.2, and 6.3. There are some terms which are not clear are not explaining on the text, so there are some terms which can be found. These are my suggestions according to what I read. I'm more from the part of modeling applied cheminformatics. So I'm not capable of making too many suggestions on the computer science part. So my focus is mainly on that part. This is only regarding the text. So now I have a few questions or things that is worth to clarify, and some parts where I would like to know your opinion. First, you mentioned that we should aim at obtaining interpretable models, but are these type of models always possible? And if you obtain one model that is interpretable, you would discard a black box that exists that interpretable model in performance, for example.
Okay, so thank you for the observations. Okay, and yes, I mentioned that the question is that there is this trade-off between the performance that you may have, that you may add to your model with some black box models, but there is this need in my criteria, in my consideration, that in the field of bioinformatics that related to health, there is this need in order to say this molecule binds to this cellular line, for example, but this is the reason. Okay, in order to be able to point the reason that it binds, I think that it is important that this is why we should aim to develop interpretable models. Okay. However, currently now we have very good black box models. So I understand that in bioinformatics, researchers prefer always performance and capability, and do not. I don't think that they do not care very much about the interpretations of the causes or the causalities, but in my understanding, this should be addressed, and this should be taken into account, the interpretability of the models. Okay, but we have a spectrum. In one end, we have a completely opaque model, a black box model, and in the other pole of the spectrum, we have a completely transparent model. So maybe if we can have a tendency, at least, for a much more interpretable, much more understandable model, instead of a black box model. Okay, so this is my criteria there, because there is not always possible to have an interpretable model, because the availability of the information maybe is not very wide. There is not much information that can be that it is available to form a database. But also, for me, it is very important to know why things happen, on why there is this prediction. So why is this drug, why is this compound active to this cellular line? I am worried about why, and maybe in bioinformatics, researchers are not very much into that. They prefer results and not interpretations. But in my consideration, an interpretation is very important in order to understand first the problem and the solution, and after we understand the problem and the solution, we can grow on top of these solutions. Okay, if we are constructing our knowledge and our decisions on top of black box models, I think, following some other expert, that maybe this pyramid will crumble. And when someone says, yeah, but why? Why do we choose this? Why do we go for this path? Why do we go from this? Why do we take these decisions? Okay, so for me, it is very important to understand why we are building these decisions one on top of the others, having clear explanations. So maybe this is my criteria, what I think, but this is my answer to this question.
Fine. This could be an endless discussion.
Yes.
So maybe we have this discussion on a bar, not here.
Okay.
So it's okay. I just wanted to know. Could you briefly, because I know this texture representation for cell lines is novel, but could you please briefly mention which advantage could you have relative to the existing representations, to pure representation?
What I'm sorry, I didn't understand. What are they?
Which
The differences.
advantages?
The advantages.
Which advantages that you can have with your representation?
What are the advantages from my work to the other types of representation? Okay, first of all, I didn't have to do any lab work in the. It was only in silico, so for me it was an advantage since I am not bioinformatics in that kind of sense. That's the first advantage. The second advantage is, as I said, the capability, the ability to do not depend on other factors that it's like this. So I try to understand what is a cellular line, what is this specific cellular line. So if you say, for example, that the representation depends on other things, okay, I am not sure if the cellular line, the entity that I am trying to define, the definition actually represents what it is. Do you understand? Do I make myself clear? Okay. So I am worried. I am worried about the representation, the intrinsic representation of the cellular line. Okay. And now how it relates to other entities. For example, in the genomic profiling, it actually the representation is how this cellular line relates or reacts to the application of a compound. Okay, so what I am representing is not the cellular line, but the reaction of the cellular line to the presence of a compound. So actually the representation is not intrinsic for the cellular line. I am not representing this. I am representing how it relates to a set of compounds or one compound. Okay, so for me it has a value that I am trying to understand what a cellular line is. Okay, so in this case, I think that has an advantage, an added value, and also that I do not depend on the disease. Okay, so the molecule. Okay, so this cellular, a certain cellular line can have different genetic profiling depending on the compounds, but I am representing the intrinsic values of the cellular lines. So for me, that is the most important advantage. And so there is also these practical advantages that I do not depend on the compounds. I do not have to do experimental lab work. These, for me, are the main advantages of my approach, of my method.
It's clear for me now.
Okay.
In chapter 4, this will be more than one question. First, why to use only the Celosaurus database for extracting the information? And second, why, if you write that Celosaurus contains information for more than 118,000 cell lines. Why the final description is provided for more than 1,200 and something cell lines?
Okay, so first, the Celosaurus was selected, the database was selected because of the pipeline. Okay, so if we want to represent these cellular lines in terms of the literature that is written about them, so there is this characteristic of the cellular line. This happens that in one paper a cellular line is named in one way, and other authors in other papers name the same cellular line with other names. Okay, they relate because they present maybe in the metadata, or maybe it's something about the transcription of the work. However, Celosaurus collects all these synonyms. This is what it is called the cellular thesaurus, the Celosaurus. So for one cellular line, Celosaurus lists all the different names or the equivalences of the names. Okay, so I can work with all these names, and I search in the search engine with all these different names. So I am certain that for one entity that has many names, I try to collect all the information that is written using any of these names. Okay, see, this is the principle that guided. It also has a very large number of cellular lines. In the web page for Celosaurus, it says that it has over 100,000 cellular lines, but it can be understand that the cellular lines that are listed there, more than 100,000, are the different names that are given to these cellular lines, and not one particular cellular line that has many names. Okay, so maybe this is the difference between 100,000 and 1,000, and so on, the cellular lines that we will work with.
No, I think that this jump from one number to another is not explained in the document. So maybe in the this revised version of the document, you can include a brief description of this was the initial number. Then after grouping according to the different names, we had this number. And then after applying the document cutoff filter, then we obtain how you go from that initial number to the final number. So once you read, you can have an idea of how were the cell lines grouping and filtering. That could be more. It could be important to follow all the process of filtering the database. This is my suggestion in this case. There's something missing in the document too. That is how many bits contain the fingerprints for computing the chemical similarity, and why you choose this particular value.
Okay, so I use RDKit for the as a library for the representation of molecules, and there are different types of CMFP Morgan fingerprints for molecules, and the parameters for the fingerprints are first the radius. That I use 2, 4, and 8 as atomic radius parameters, and also I use 1024 bits because it has 512, 1024, and 2048, if I'm correct. So, for the calculation of the fingerprint, if you have a larger vector, okay, it will take more time, okay, and if you have a shorter, it will take a shorter time, a smaller time, okay. But the description for the molecules, if you have more information, it is richer, and if you have less information, it is less rich. Okay, so I thought that 124 it was like a good trade-off between the capability of description and the computing time of the fingerprint. So okay, I try to balance these two things, because since we are working with a lot of molecules, sometimes the experiments run too long, maybe a few days, maybe a couple of weeks each experiment. So if I change something like the time that it takes, the execution time of the calculation of the fingerprint, it can save for me several hours of computing time, of computing power.
It's a balance between accuracy and threshold, and accuracy and processing speed.
Exactly.
For the calculation. And you can also include that in the text to be more specific in that part. Do you have any reason in section 6.5 where you present different training and test sets with Kimbial 24 versus 25 to 29? There is a decrease as the version of the database is newer. Did you observe any reason why this tendency to decrease when relative to the case of the 24 and 25 version of Kimbial?
Okay, so okay, it's like this. I trained my model using version 24 and tested with version 25, 26, 27, 28, and 29. Okay, so the best performance is between 24 and 25, and the poorer performance, the poorest, was from 24 to 29. Okay, so the reason that I can point to is, first, is a practical reason. There are more records in the database, in the difference between 29 and 24, and the 25 minus 24. So given that I am trying to predict or assess the prediction to a larger test set, it means that it will have a poorer performance. Also, I try to understand like this: if you have an information and try to make a prediction, it is easier for you to predict something that is close in the future than something that is far in the future. So if you are in 2016 trying to predict what will happen in 2022, it is harder than if you are in 2016 and trying to predict something that will happen in 2017, for example. So this distance into the future that you are trying to predict, I think that it is the reason that it has a poorer performance between 24 and 28 versions of Campbell database.
One question. Did you compute the diversity of the chemical space in these test sets? Because I think that that could be related to this decrease in performance.
No, I didn't.
I recommend to do that for future works, so you can have a track of how many new scaffolds, for example, are included in newer version of the database, and being that new information, you will not have analogs in your reference search space to predict them. That could be another reason. So this is something that I recommend that you perform in future works related with this topic. It's just a recommendation.
Thanks.
Also, well, you are also clear that similarity methods for making predictions require high similarity between the query and the reference space. But do you have any suggestion or any idea on how could you somehow try to address this problem?
I'm sorry, I don't understand what problem.
When you make similarity searches,
Okay.
you have a query and you have the database where you perform the search with the annotated data. And similarity methods are highly dependent on finding close analogs of your queries in that database. But can you think on other methods or other ways to overcome this problem with similarity methods?
Well, what I try to do is to have this dependence on similarity to diversify this dependence. Okay, so in one method you depend on the similarity between molecules, in the other method you depend on the similarity between cellular lines, for example. And if you combine these two, the failures, the disadvantages of each can be overcome when you combine these two methods. Okay, so my strategy was actually the consensus. So if we have a consensus, we can leverage one method to the other, and in this case we can overcome these difficulties of these methods. So for me, it was this strategy. I'm sure there will be some other strategies, but that I am not aware of.
Have you considered representing the compound, for example, not only using chemical fingerprints, that for me is the description that limits this high similarity between queries and reference compound? Have you tried to extend the restriction of the compounds with other types of molecular descriptors, for example?
I am not sure if but I am not sure how to combine these descriptions. So for example, I use fingerprints and other descriptors, and sorry, you combine them.
My mistake, not combining, trying another descriptions.
Well, I was doing my research and my thesis. I didn't think of this. For me, it was in the molecular aspect of this problem. For me, it wasn't, like, how can I say? For me, it was clear that since I have read a lot of work that relies upon the molecular descriptors, the fingerprint descriptions, so for me it was like a de facto standard for the. Yeah. So if everyone does like this, so what? I should try the same thing. Okay, for the cellular line representation, I wanted to focus on that problem and to try these approaches and these strategies for the representation of cellular lines, but it didn't occur to me that I could use another type of descriptors. Of course, that there are also other ways to represent molecules, but it didn't occur to me that it can be done.
No, but I mean, fingerprints are not a bad representation. No, not at all. But they have this limitation with the similarity. You can jump to dimensional descriptors, continuum descriptors, and you will find another problems. But maybe since you already are working in these ensemble models, maybe you can try models with different types of descriptors, which could serve also a base learners. And then you can have different outputs. You should have the diversity in the outputs if you use different descriptions. So this is something that you could try in the future. It's just a suggestion too.
Okay. There is a lot of work to be done in the future.
It's not bad. It's not bad to use only fingerprints. It's just a suggestion that could improve the accuracy of your and the performance of your final model. Okay. So what else? It's something that I'm curious about. The threshold that you establish in section 7.4, this minus 0.43, that value is closer to non-interaction between compounds and cell lines. How can you interpret this result? Okay, so
can I share my screen? Can I? Let me.
Yeah.
Okay. Okay, you are referring, I think, to this one.
Yeah, you had the graphic.
This one.
This one.
Okay. Yes, I think you're referring to. Okay, so my first interpretation is that I should have done two approximations. So I have here one threshold. Okay, so I have these three parameters, these performance metrics: true positive, false positive, and accuracy. And the best accuracy is accomplished here. Okay, so in minus 0.43. Okay, but here's the thing. I think that I could change these approximations and have two thresholds. One threshold to approximate any value from minus 0.43 to minus 0.99 to minus 1. So the approximation is to the left in this case. Okay, and as you see, the accuracy almost that it is constant up until this other value 0.51. Okay, and then decreases. Okay, the accuracy decreases. Okay, so maybe close to here I have another threshold. Okay, I have another threshold for the values that can be approximated to 1. Okay, so if it is bigger than 0.51, I can approximate to 1. Okay, and I have three values: minus 1, the values that are from minus 0.43 to minus 1. This is minus 1. This is 0 to be unknown, for example. And plus 1, okay, because the other works, compound fishing and cell fishing, have these three values: minus 1, plus 1, and 0. Okay, so I think I can improve this model like this. Okay, this is a hypothesis that I had a mistake having only one threshold and having two values for the recommended system. If I can have two thresholds and three values for the recommender systems, maybe I can improve, and maybe it makes more sense to have two thresholds than only one.
Yes, yeah, that probably could fix this. I mean, this is something that I cannot rotate at the beginning because of this value being closer to non-interacting than to interact. I mean, there is something you have to work with. I understand. Uh, Finally, how much time do I have left? Oh, almost 10 minutes, I think.
Yes,
Just one
about 10
more.
minutes. Yeah.
Yeah, just one more specific question. It's related to the ensemble models. In the table that you present with the accuracy of the ensemble models, there are less predictions for the cell fishing and the compound fishing models. So I know these models are more restrictive in terms of